Hello and welcome to Nikolay's genetics lessons, and today we are going to talk about gel electrophoresis mistakes. And here's a question I got from one of my subscribers: why does my agarose gel look like this? And specifically, why do my PCR products look like this on my gel? The products are amplified using lamp specific for E. coli. Lane 1 contains 1 kilobase ladder. We do not see anything here. 2 contains E. coli, and 7 contains negative control. So next, lanes 3, 4, and 5, and 6 contain bacteria that are not E. coli. I used 1.2% agarose gel and 1x TBE buffer. Actually, what you see here can be a result of not single mistake, but multiple mistakes. So I'm just going to list all of them. Number 1 reason for gel electrophoresis like this, as you see, it is obvious, far from being ideal, can be voltage fluctuations. So you probably cannot do much about this unless you're using special equipment that reduce such fluctuations, or you just probably have to redo your experiment. Reason number 2 can be improper preparation of the agarose gel. Improper melting or casting of the agarose gel will lead to ununiform concentration of the agarose throughout the gel, and this is probably can be one of the main reasons here. As you see, it looks like here we have uneven gel. Another reason for such a gel can be above or bottom leakage. In order to avoid above leakage, don't fill your wells more than 2/3 with your sample. As for the bottom leakage, you also can see the same picture as in above leakage, but the reason would be different. It may be that your gel comp is too deep into the agarose, and the bottle of the gel can be splitting when you remove the comp, allowing sample mixing while loading, and the sample is running under the gel and not through it. How to avoid bottom leakage? You have to try cleaning the comp well and putting a spacer under it to lift the teeth higher into the gel, and look for samples spreading before the electrophoresis is started. I would also recommend to check your PCR machine. Heating block may not be working properly. I also think that there can be a problem with PCR reaction preparation, such as DNA contamination, old chemicals, forget to add any of the components, or contains PCR inhibitors, ethanol not removed properly as last step of DNA isolation, wrong PCR conditions, and so on. Especially looking at these bright dots here, which is all over the surface of this gel, we can say that probably chemicals was not properly mixed. And lastly, I would also check buffer concentration and pH, because even DNA ladder is not visible. You may need to rerun and see if the ladder is well resolved or not. If not, buffer concentration, pH may be a culprit. And this is all my advices for today. Subscribe and see you in the next video. Goodbye.